Hello everyone, we're here in this lab today with Heather and Anna. We're really excited to be here, and they're going to show us their lab and tell us a little bit about the researches that they're coming up with. So they're going to give us a tour today. Thank you so much.
Yeah, no
Let's
problem.
start with the tour.
Yes, of course. So this whole area is our lab. It belongs to Dr. Timothy Kiefer, and we all work together on individual projects that are working towards the same goal of finding new treatments for diabetes, both type 1 diabetes and type 2 diabetes. Okay? So each of the individual researchers, we get our own lab space here, where you can see we have all the things we need to conduct our research projects. Right? And then we'll go through here, show them. We have a number of smaller research areas that we have different equipment in. So for example, back here we have a microscope connected to a computer, and we can image things like pancreas sections to look at different hormones and their expression levels in there. Here we have a number of more complex technologies. So we do a lot of hormone assays here. We're trying to understand how insulin can interact with other hormones, or even hormones themselves can promote or preserve beta cell function and prevent diabetes. So we use these kinds of equipment to do that over there. Yeah, here we grow bacteria. So this is for cloning genes, particularly.
We were just learning
genes. Yeah, so we clone a lot of genes that we believe are related to diabetes in order to study the function of those genes for the gene products. So that's basically happening right there in that vial there. You can see it's getting filled with bacteria that are carrying the gene of interest. And sometimes when we want to do things like high throughput, where we have thousands of samples we need to assay at once, it would take a long time for us to individually assay them. So we have this robot here, and basically what it does is you can have multiple wells that you are assaying at the same time. So you can significantly reduce the amount of time you need to spend on it.
That's cool.
Cell culture. So in cell culture, we use different cell lines to try to understand how hormones can either prevent or cure diabetes. And here we also, so we grow a lot of different types of cells, particularly a lot of different types of cells that can become beta cells. But they require a lot of work to keep all the time. You have to feed your cells, you have to care for them, make sure they don't get contaminated. And it's a lot of work. So what we actually can do is, this tank is filled with liquid nitrogen, super cold. So we can basically cryopreserve our cells. They'll last forever, like this in here, and then when we need them, we can take them out, thaw them, and they're as good as new. So
Research on them and whatever you want.
Exactly. Cool. Yeah. So we have all our valuable cells at a cold temperature, because say certain proteins are really susceptible to degradation at higher temperatures. So we have this big, gigantic fridge.
Wow.
And it comes, and you can feel how cold it is. So sometimes we have to work in here all day, and you have to bundle up.
Yeah.
It's quite cold.
So this just
Are these like all chemicals?
These are, basically all chemicals, different reagents that we need, different samples that we want to preserve. Because they are hormone and protein based, we keep them in here before we use them.
So the work that you guys have to do with those proteins, do you like do it in this room or do you like take it out?
It depends on the work. So we do some of it in here and some of it in this. So we'll show you this poster. Now this doesn't include every single thing that our lab is doing, but it has sort of a major overview. So our lab works on both type 1 and type 2 diabetes. With type 1 diabetes, we're really focused on how can we develop cures for it or better treatments. So some of the major things we're doing is, one, we really want to be able to image the development and understand how the insulin-producing cells in our body develop, because that can tell us a lot of answers about how they can be attacked by the immune system, and also how we can mimic that development to make our own beta cells. So we do lots of things like putting genes that encode fluorescent proteins into beta cells, and then using microscopes we can track the development of these cells over time. Another major thing that we do is we're trying to take stem cells and convert them into beta cells. So this would allow us to have a source of beta cells that would never run out, and in theory we could treat all people with type 1 diabetes with the transplantation of these insulin-producing cells. So that's a major aspect of our lab as well, is developing how do we mature stem cells into insulin producing beta cells. Another aspect is, okay, instead of making cells in vitro that can express insulin, can we target the insulin gene to cells of our own body? So other than beta cells, because after you have type 1 diabetes, those are already gone.
Yeah.
Can we find another cell in the body that can take the insulin gene if we give it to it
Yeah.
and express it and secrete insulin in an appropriate manner?
Yeah, because we have to take insulin for certain diabetes, right?
Yeah.
Exactly.
Which
Yeah.
is the
For,
fun.
yeah. Exactly. And it has major limitations
Yeah.
in terms of how well it can control blood sugar.
Yeah.
So you can do that better if you have a cell that can sense glucose levels
Yeah.
or blood sugar levels and
Yeah.
release the appropriate amount of insulin at the exact appropriate time.
Yeah.
And so another way to do that is we've been trying to engineer insulin that can be delivered to cells within the gut. So they line our intestine, and they're called K cells.
Yeah.
And these cells actually, they secrete a different hormone,
Yeah.
but they secrete that hormone in the same way that a beta cell secretes insulin in response to blood sugar. So these cells also sense blood sugar and secrete it in the same way.
Okay.
Can we sort of take those cells and hijack them and make them secrete insulin instead of what they normally secrete? And that's one of the things we're trying to do. Yeah.
So the other arm of our lab is to understand a little bit more about type 2 diabetes. So type 1 diabetes, it's well known that it's caused by autoimmune destruction.
Yeah.
But type 2 diabetes, the causes of it are less well known. So our lab is particularly interested in a hormone called leptin. So with type 2 diabetes, there is a high prevalence of individuals who are obese. When you're obese, you have more fat cells. And
Yeah.
with more fat cells, you have more of leptins, because leptin is produced from fat cells. So we're trying to understand how leptin can either act on its own and maybe help in alleviating diabetes, or interact with insulin and provide a form of therapy for treatment of type 2 diabetes. So a form of looking at this is to look at what would happen if we take away leptin, what happens to these animals. So this, you have two types of mice over here. So these mice have leptin, and these mice don't have leptin. You can tell the mice that don't have leptin are really obese.
Yeah.
And not only do they get really obese, but they also get really diabetic.
Yeah.
So we're trying to figure out, okay, we know that leptin could potentially be important for treatment of type 2 diabetes, but how is it doing that? So that's what a lot of our projects focus on. Is it
Okay.
through direct mechanisms of leptin and other tissues, or is it through leptin acting on other hormones? And that's what we're trying to figure out here.
But now more and more children are becoming obese, and it's because globally, we have a change in the type of food we eat. So we
Yeah.
like McDonald's more and more,
Yeah,
and
that's true.
we play more video games, and we
Get
go outside
lazy.
and run less, right?
Yeah.
So you have more and more children that are developing obesity, and now more and more children are actually getting adult onset diabetes. So they changed it from adult onset diabetes to type 2 diabetes.
Okay.
Yeah.
So like, which one is more common, type 1 or
Type 2 is definitely more common. So about 85 to 95% of individuals with diabetes have type 2. And that again is related to the fact that it's mostly due to having too much food and not enough activity available. Type 1 diabetes is more related to your autoimmune function.
Okay.
So although both there are genetic links to it, type 2 is definitely more susceptible to environmental factors that can contribute to having a higher prevalence of it.
Can like type 2 diabetes be cured right away? Because then it won't target type 1 diabetes? Because you guys are looking into the cures, right?
Yeah.
So that's my question.
So we don't want to call it a cure necessarily, it's more of a therapy.
Oh,
So you guys are trying to find out?
Yeah, we're trying to find ways of managing the diabetes so they don't progress further.
Oh.
But once
yeah,
you have, for example, destruction of beta cells, you can't really undo that. You can only prevent it from going further.
Okay.
Yeah.
So basically, in other words, will there be a cure? And at one point, like, I don't know, 40 years, 30 years, I don't know how many years.
Yeah, it
We believe
depends.
there will be.
It depends
Yeah.
on the type of diabetes that we're discussing.
Yeah, I mean, I would say, so type 1 diabetes, we discovered that people with type 1 diabetes are lacking insulin over 90 years ago. That's when the hormone insulin was discovered, and it was actually a Canadian discovery. So we have been working on type 1 diabetes for a very long time, and we know quite a bit about it.
Yeah.
I would say within our lifetime, we could probably find a cure for type 1 diabetes. And that would be there's sort of two strategies. One, if we could, so the beta cells have been destroyed
Yeah.
already, can we put cells back that can produce insulin
Oh, yeah.
and do it in a regulated manner?
Yeah.
So strategies like that could be a potential cure, and that could be growing stem cells to produce insulin and become beta-like cells.
Yeah.
That could also be, we currently do islet transplants from donor tissue. We take those islets which contain the beta cells and transplant them into people.
Yeah.
The problem, so that is actually really promising as a cure for type 1 diabetes.
Yeah.
The problem is there's not enough donor tissue to go around.
Okay.
So really a strategy to find another way to have a never-ending source of beta cells would be a cure for type 1 diabetes.
Okay.
Another strategy is because type 1 diabetes is an autoimmune disease,
Yeah.
so your immune system mistakenly kills all the beta cells.
Yeah.
Other labs are working on, can we prevent that autoimmune destruction before it even gets to the point where the beta cells are dead?
Yeah.
That would also be a cure for type 1 diabetes. Yeah, and we're getting closer and closer, I would say.
So diabetes basically just opens doors to more diseases.
Yeah, stroke, heart attack, blindness, limb amputation, kidney failure, all of those things. So the longer you have it, the more devastating it can be, for sure.
So you basically can't get rid of it.
No,
No.
so that's why a large portion of research is dedicated to therapies to prolong your quality of life,
Basically can
where you're
kill them.
disease and complication free.
Okay.
So approximately how long do people live with diabetes?
Yeah, like the lifespan.
Yeah.
So
Approximately.
average lifespan is shortened by about 15 years with diabetes. Now it depends on how well you're able to control your blood sugar levels, and that's not always the person's fault. Sometimes if they're more prone genetically, or they've had a bigger environmental insult to their body, they just can't control their blood sugar levels as well. So that chronic high level of blood sugar will really be damaging over time, and the average is a reduction in 15 years, but that can be changed with how well you control your blood sugar.
In this building, have this general structure where it's a general open lab space. Earlier in older labs, there was lots of closed doors, and it wasn't very facilitative to collaboration.
Yeah.
But here we have this huge open space, and down there's another lab, Jim Johnson's lab. He also does diabetes research, so it allows for a lot of collaboration between our labs, where we can share chemicals and equipment and things like that.
Yeah.
Okay.
Can really speed up sort of the progress of science. If we're all sharing at the same time as
Yeah.
we get results,
Or
then it's
like,
much faster.
Yeah, and they probably have like the leading topic as well that can connect to that, right?
Exactly. Yeah.
Is there like certain equipment that you really need for that diabetes research?
That's a great question. I mean, there's
Research is important.
which is definitely lots of people need the cell culturing, because a major part of diabetes research is trying to manufacture beta cells from stem cells.
Okay.
And so you definitely need cell culture equipment.
And we also use a lot of imaging technology.
Okay.
So we have the ability to take a pancreas and we fix it so basically none of the cells can degrade, and then we can stain it with different antibodies. And so these antibodies can recognize, for example, the insulin cells and the glucagon cells. And from there we can understand how the disease progresses, or how we can achieve a cell type that's more close to a beta cell.
Yeah.
Exactly.
Yeah,
so we'll
so
take you to
microscopes.
that room
Okay.
later on.
Okay. So
Let's go that way. We have a number of computers and processors dedicated to imaging either cells or slides that have pancreas sections on them. So each of these computers is able to basically take little snapshots of a section of a pancreas, and then we can zoom into it really close and look at these individual islets. To the naked eye, you wouldn't be able to see these islets. So that's why when we do this slide scanning, we can take little pictures, stitch them together into one big picture. Think of it like Google Maps. So Google Maps, you can see the your entire city of Vancouver, and then you can zoom in specifically to your street and see how your street looks like. Well, this is the same thing here. We can take an entire picture of a pancreas, and then we can zoom into a particular islet here. So this is an islet, and look at that. Compare it to an islet from another different type of treatment, and see what the differences are.
just islet itself.
So the islet is basically the functional component that produces either insulin or glucagon for blood sugar regulation. Yeah. So within our pancreas, the islet basically is a little island, and on that island you can have beta cells which produce insulin, you can have alpha cells which produce glucagon, and you can have a bunch of other different types of cells that can regulate the hormone secretion. So if you look.
Okay.
Okay, so how long have you been researching on diabetes?
Like years.
Yeah.
So I've been researching diabetes for 5.5 years now,
Okay.
and that's towards achieving my PhD degree in hopes of one day going and starting my own diabetes lab.
Yeah.
Okay.
And I'm in 2.5 years into my PhD.
Oh.
Did you guys choose this topic because you're interested in it, or did you guys have like a background to it as well?
Well, so like you guys, I have a lot of family members with diabetes, type 2 diabetes,
Yeah.
and it has to do with the genetic and ethnic link. So that's why I was always interested.
Yeah, and I actually had a couple friends who were diagnosed with type 1 diabetes,
Yeah.
not as children, but actually as in early adulthood. So that sort of made me interested in
Yeah.
diabetes research.
Okay.
I mean, so insulin was discovered in the 1920s, and diabetes had been researched long before that as well.
Oh, okay.
But
So there's so many like I guess like preventions to it now.
yeah,
Yeah. Like insulin and pills and everything. Because I don't think that was back then, like 1920s.
Right.
yeah, they used to purify insulin from animals, and it was actually so impure that it was this brown mixture, and they would inject it into people. But the important thing is that those people before insulin would die almost immediately of diabetes.
Diabetes
And-
was considered an acute disease. So if you were a child and you had diabetes, you wouldn't really live past childhood. But once insulin came into the picture, now individuals were
live
living
longer.
kind of normal lives
Yeah.
Yeah.
up until late
Well, yeah,
adulthood.
because when people say we have diabetes, it's not a really big shock because I guess it's not really considered a big disease now because there's so many things you can do with it.
Yeah,
Yeah.
and a lot of people assume diabetes has been cured with
Yeah.
insulin, but that's not the case. It's
Yeah.
a treatment that's changed it from an acute fatal disease to a lifelong manageable disease.
Okay. Yeah, I've heard about type 3 diabetes. Yeah, what is type 3?
What have you heard about type 3 diabetes?
Like, it's just like a new.
Diabetes that's coming, that's
Yeah.
what we heard, but we don't know what it is. Like,
Yeah.
what's the new diabetes? What is
So
diabetes?
type 3 is basically the new trend where they focus more on the link between Alzheimer's and diabetes.
Oh.
So
okay.
within the brain, you can have insulin resistance. It's very similar to type 2 diabetes. But the more striking fact is that a lot of individuals who have Alzheimer's also had high incidence of type 2 diabetes. So that's why they're suggesting that type 3 could
Could be a
be
connection.
another form of diabetes.
Okay, yeah, thank you so much. And we really appreciate you guys helping us for our project and you taking time to help us.
We appreciate you guys
Yeah.
coming.
Thank you so much. It's fascinating.
Thank you.
Thank you. Thanks.